Hello, everybody. Welcome to BioacousTalks. It is currently a sunny day here in New York, and I'm extremely happy about that. So please give a shout in the chat where you are saying hello from. And today we are welcoming morning person extraordinaire Thomas Napier, who is joining us from, 6 a.m. in sunny Australia to tell us about his a new tool that he built in the bioacoustics arms race. Thomas has created a way to automate the annotation process and maybe saving 7 to 8 times the time that it takes us to normally annotate. And we all know how consuming a process that could be. And he's currently, I think, almost towards the end of your PhD, right? And is coming from computer vision and marine biology and is bringing some different perspectives and tools and now applying them for bioacoustics analysis. So he'll share the tool, give us a demo, and then really as he's going, think of some questions. If you're interested in using this tool, at the end of the presentation, we'll have about 15 minutes for Q&A. And as always, we have a Q&A box at the bottom of our Zoom, and you can also translate in real time to over 24 languages by clicking the CC button and then choosing what the caption language you want to be. And with that, Thomas, really very warm welcome to you and looking forward to hearing about what you've been working on.
Thank you so much, Ben, and hello everyone. Thank you for joining around the world. It's an honor to be presenting here today. Let me just go ahead and share my slides before I begin. So my name is Thomas Napier, and I'm really excited to speak with you all today about the future of passive acoustic monitoring and sort of how we can scale up our analysis workflows. And it's a real pleasure to be sharing some of my work with this community of bioacousticians and PAM practitioners. It's not always the case, especially where I'm from, a small town in North Queensland, Australia, that I can always have a sort of audience. So yeah, it's a privilege to be here today. So before I sort of dive in, I just wanted to give a brief sort of roadmap of what I'll cover in today's session. So I'll begin by sort of going over the sort of introduction to the area and a little bit about myself, and I'll move quickly on to sort of how we get the PAM pipeline as it currently stands and some of the major challenges, sort of how we go from raw audio to ecological insights, and I'll highlight some of the pain points, especially in the sort of segmentation and annotation area, as those tend to be the sort of bottlenecks. I'll then move on to the current approaches and cover some successes and gaps. So I'll look at a few different tools we have out there and methods for different ways to analyze our acoustic data. Spoiler alert, we have great classifiers, but we're still drowning in unlabeled data. So I'll introduce my new tool, the Leaves tool. It's a platform for ecoacoustics annotation, hopefully designed to tackle some of the bottlenecks that I'll cover, and I'll introduce some of the key innovations and how it can improve the annotation efficiency by up to 7 times over manual methods. I'll hopefully go through a live demo, a realistic sort of live demo, showing you how to load, sort of how the tool sort of works, focusing on how you'd actually use it in practice, and then I'll sort of wrap it up. So Just by way of background, a little bit about myself. I'm in the my final year of my PhD in information technology at James Cook University in tropical North Queensland, where I've been for the last few years developing new methods for handling large-scale ecoacoustics data sets. And so, we're pretty lucky here in Australia. We've got some quite incredibly rich and beautiful natural habitats down here, all the way from tropical rainforests to sort of bushland. And to be honest, that's sort of what inspired me to sort of get into ecoacoustics. So I've always wanted to find ways to help sort of protect these things and sort of natural, beautiful things that we have here in Australia. And so, like Ben mentioned at the start, I came into ecoacoustics from a marine biology honors project. And so this sort of felt like a natural step that let me combine my passion for wildlife with skills in software development and human-computer interaction. So a lot of the work I've been doing has been focused on building tools that improve efficiency, because I genuinely believe that if something can be done better, it probably should be. And so that's sort of the philosophy that led to Leaves, which is the tool I'll be showing you today. So enough about me. So the question is why, why, before I get into sort of how Leaves works, I want to take a moment to sort of zoom out and really try and delve into why this kind of work matters, especially here in Australia, which is home to over 500,000 different species, including many that are endemic to this region. And so, with so much unique biodiversity under threat, not just in Australia, but worldwide, improving how we monitor and protect these species is more important than ever. So, as I'm sure many of you know, passive acoustic monitoring has obviously revolutionized how we study wildlife. And, we can now leave out autonomous recorders for weeks or months, continuously collecting sound data for us. And so this gives us obviously great temporal and spatial coverage. However, what we're seeing now is sort of the challenge has sort of shifted from collecting the data to now making sense of it. And so, we've got terabytes of recordings and now turning those into actually meaningful ecological insights is a major bottleneck. So one of the big problems is that, obviously with traditional monitoring techniques like field surveys or manual audio reviews, these things are obviously pretty labor intensive and really don't scale with the volume of data that we now collect. So as a computer scientist, it genuinely pains me to see just how many recent studies still rely heavily on manual validation as a core part of their methodology. These are a few of the papers, just recent papers I found, and they're all high quality and there's no criticism there, but it really highlights how limited our current tools can be when it comes to scaling analysis. Despite all this talk of automation, we're still, at the end of the day, asking humans to, click through a spectrogram one by one to clean up the outputs or confirm the species IDs. And so, obviously I get it. We need expert judgment to maintain confidence in our results, but the fact is that this step is still so common that it shows either our current tools don't scale, they don't generalize, or they're simply just not trusted enough to sort of stand on their own. So this leads me to sort of the driving force behind my study, which is the Australian Acoustic Observatory, or the A2O as we call it. So, when we start looking at these sort of continental-wide initiatives, like the A2O in Australia, or obviously, I think in the US, you've got the Northeast Passive Acoustic Sensing Network. I think it's called NEPAN. Obviously, when we start looking at these sort of networks, the challenges with manual validation just become even more extreme. And, we're no longer talking about just a few recorders over a few weeks, because as far as I'm aware, the A2O alone has over 360 autonomous recorders across 90 different sites running, 24/7. So at this scale, manual approaches just simply aren't feasible. So the only way to keep up is to start seriously thinking about automation. And by the way, I'd highly recommend checking out Professor Paul Rowe's BioacousTalk, where he covers more about this A2O and sort of how he was one of the sort of main contributors behind that. So he talks a lot about how he sort of put it together. So if you're interested in that, I'd highly recommend looking at his BioacousTalk. So yeah, data collection is where things really start to diverge. We've got this data collection has started to massively outpace our capacity to analyze it. So just from the A2O alone, like I was saying, we're seeing an increase of 3 million hours of audio every single year, and that's not obviously not including other networks or individual monitoring studies, or that's just in Australia alone. So the problem is, whether it's manual annotation or even some automated tools, they're still too time intensive and often require either a level of technical or taxonomic expertise that not every research team out there has. So we're sort of left with this growing backlog of incredibly rich, under-analyzed data. And unless we find a more sort of efficient way, more accessible ways of working with it, we're at risk of losing the value of it, because of the time. So this sort of brings me to the sort of current pipeline that we have. And I took this straight from my survey I published last year in the Journal of Expert Systems with Applications Journal. So on the sort of far left, we begin with the raw audio signals direct from the recorders and that sort of thing. Obviously, this is based on the sort of survey design and that sort of thing. But once we sort of have the raw data, obviously things like data handling becomes a stage where we have to look at things like formatting, storaging, and that sort of thing. Once again, Professor Paul Rowe covers that nicely in his BioacousTalks. Next step we have is pre-processing, which can include a bunch of different things like signal denoising, bandpass filtering, other transformations of that kind. These processing steps are largely modular and in many cases optional, depending on the goals of your analysis. Similarly, we have a sort of optional visualization stage where it can sort of help us to provide a sense of structure from across long periods of time. Here you've got things like false color spectrograms and all these sort of nice things that help us explore days or weeks of data in a digestible format. But when we get to right here, this is when things get tough. And where I've particularly been focusing a lot of my own research is right here. So the sort of transition from when we've got this pre-processed data to the sort of labeling and segmentation is where I find a lot of friction piles up. So I define segmentation as where we sort of break down the continuous audio into meaningful segments, which could be an individual call or a sound event that we can then analyze and classify. So segmentation alone is, where we decide where the beginning and end is. And I believe it's actually a hugely under-discussed area, yet I believe it's foundational, because if you have poor segmentation, then that cascades into poor classification and then ultimately, weak ecological conclusions. You can imagine splitting calls into pieces, or multiple species sort of getting mixed all into one segment. So these are all problems that can affect the sort of downward stream. The other one is we've got annotation, where we try to label those segments with the species or the sound source, depending on, again, the sort of study, right? So sometimes it could be a broader category, like biophony or geophony, that sort of thing. So I guess the key question I want to raise here is, how can we reduce this friction, and how do we scale up these workflows? Because it's clear they're not really working for the sort of large scale data we're seeing. So there's really, two main challenges that I believe we need to really focus on as a community. And the first is open ecoacoustic monitoring. So I firmly believe that we need open source, transparent platforms that sort of the broader community of researchers, citizen scientists, land managers that can actually use and contribute to. I find that too many of these sort of analysis pipelines today are locked behind either some custom code or black box tools, and that sort of limits the scalability and ultimately, I think, the trust. So I think community-driven tools are essential if we want to maintain this sort of reproducibility and shared progress. The second major problem we have is robust sound distinction. So, natural soundscapes are obviously heterogeneous and unpredictable. So, in a single recording, you might have multiple sound sources overlapping. You might have like a bird song with an insect chorus in the background, and then a gust of wind all at once, depending on your sort of segment length, right? So overlapping calls and noise make it hard to decide what is a segment and which label applies. Many, from what I've seen, many current methods have sort of been developed around, relatively clean or sort of curated data sets, and so they sort of struggle with this messiness of the sort of real soundscapes, which obviously include the interplay of all these, layers of biophony and geophony and anthrophony. So I think ultimately, to automate segmentation and classification reliably, we need to accurately distinguish overlapping sounds, especially in really cluttered or complex soundscapes. And again, if we don't, it suffers from, everything downstream suffers. So despite these major challenges, there have been obviously some really exciting developments across the sort of PAM analysis pipeline in recent years. And so I'd just like to highlight a few. So likely, you've probably been using, many people here have been using, things like BirdNet and, acoustic indices and these sort of things in order to sort of address the problems. But, especially with BirdNet being a sort of prime example for bird calls. And obviously, as well, on top of that, is you've also got other researchers who are developing their own classifiers for target species, using a random forest or CNN model trained on a known call for a particular frog or bird. Acoustic indices are, from what I can tell, are becoming, fairly popular for a quick assessment of soundscape patterns and biodiversity. And then the sort of last approach that, that I've really seen out there is the unsupervised clustering, where you sort of let the data speak for itself and you sort of find repeating patterns or clusters of similar sounds without any prior labels. And so I believe that this is sort of the way forward, and it can help with both segmentation and annotation. But I'd just like to briefly go through some of the strengths and gaps for each of those sort of three main areas. So for the supervised models, obviously one of the headlines being BirdNet, and these sort of models, they take in audio input from large soundscape data and output probabilities for species present. And so these tools have dramatically increased our ability to detect bird species in large audio data sets, but even achieving up to expert level performance. And I'm sure many of you have tried BirdNet, and it's a really good example of sort of academia and AI sort of translating into practical tools. So one of the big strengths of BirdNet is that it's made the process of it's made it feasible to process huge data sets and automatically tag bird species. And so just that alone is already represents a massive stride without requiring an expert to hear a recall. But unfortunately, despite the convenience of these classifiers, it does have a few caveats. The first is that BirdNet is only as good as its training data. So BirdNet knows obviously a lot about North American and European bird songs and some other species, but if your audio data has some uncommon species or sort of other taxon like frogs or insects, for example, it might not be as well recognizing them. So the second is that accuracy can vary. So these models can produce false positives or miscalls in noisy conditions, and this sort of goes back to the point of robust sound distinction, I believe. And so the final sort of point here is that in practice, from what I can tell, is that a lot of these automated results still often need post-processing and sort of expert review to ensure reliability. Even so, I think the bottom line is that these classifiers greatly speed up analysis, but they still don't eliminate that need for human expertise and oversight. And so the way I see it is we sort of become more like reviewers or editors of the AI output rather than doing all the identification from scratch. So enough about supervised stuff. So acoustic indices. So indices are a really straightforward way of and fast way of calculating, even on long recordings, providing ways to reduce audio into manageable sizes and manageable numbers. So it tackles that sort of scalability issue that I think we're experiencing. However, even with that, we've got some really nice visual summaries as well. So things like the long duration false spectrograms, which if you haven't, I'm sure most people are familiar with this, but by Michael Towsey, it's sort of a nice visualization of how sort of looking at the acoustic activity across a day or across 1 minute intervals, for example. Unfortunately, there's some gaps with indices. So from what I can tell, they are not definitive. They give clues, but they still require, again, that human interpretation. And then specifically and importantly, they don't directly label the specific event or species. So, you might see an interesting pattern in a long duration full spectrogram, but then you have to still go back to the audio and figure out what caused it at the end of the day. So I think ultimately these methods and tools, acoustic indices, they help to reduce the data volume, but they don't really reduce that need they don't eliminate that need for expert analysis. Another thing is that they can also miss, rare events depending on how the sort of the indices are being calculated. But overall, they're great for sort of triaging and flagging where interesting sounds might be, but they're not really from my perspective, not really a complete solution to the annotation problem that we have. And then we've got the final one, which is, given this sort of bottleneck that we have, I think the most promising is unsupervised learning, which is where we let the algorithm find the structure in audio without providing any labels. So one strategy is that we might automatically segment the audio into short snippets, could be a few seconds long, And then we use some sort of feature extraction and clustering to group similar snippets together. And so the idea is that once sounds, that are acoustically similar, potentially that are the same source or species will cluster together, allowing hopefully an expert to label that cluster as a whole instead of each snippet individually. So what really works with unsupervised clustering is that it directly attacks the labeling problem by reducing human workload. And so it sort of leverages the inherent structure and redundancy in acoustic data, and it finds these and lumps them together, and this is shown to be effective for sort of grouping sound events together across sites with minimal human intervention. And another great thing about clustering is that it isn't limited to known species. So, like how with BirdNet, for example, it has to be trained with a large amount of data, labeled data, in order for it to be able to recognize that new species. With this, it can just sort of discover or unknown or unexpected sound groupings. And so what I find is that this is really great for ecosystem level analysis, where you might not have a predefined category for every sound. It also naturally, this includes abiotic sounds. And so, if there's a recurring pattern of wind or rain or that sort of thing, it will form a cluster. And so that can actually be helpful in sort of broaden our scope beyond the handful of species that a classifier might be trained on. So a few caveats again with unsupervised. They're not magic. Often they produce coarse clusters that need, again, interpretation. So you might be noticing a common theme here. So, you might get a cluster, for example, that is mostly insects, calls, but with a bit of high frequency noise. And so it still takes an expert to decide what to call that cluster and, maybe split it up if it's further, if it's mixed, for example. There's also the risk of misclustering, which is where sounds that are different but acoustically similar, a frog and a bird with similar tonal calls, for example, which is quite common in Australia, where they might end up clustering together. And so that's another problem that unfortunately we have with unsupervised clustering has. And so you can't just blindly trust it. The final thing is that it can sometimes be computationally expensive, especially extracting features from terabytes of audio and then clustering those tens of thousands of points can be heavy. But luckily, we're getting faster and faster algorithms and better feature normalization and better use of sampling strategies. So all of that to say it's becoming more feasible to apply this to continental scale data sets. So in summary, clustering offers a way to sort of tackle the labeling bottleneck that we have by letting the data organize itself, and then we involve humans strategically rather than sort of indiscriminately. And this is exactly the philosophy behind the Leaves tool, which I will introduce now. So Leaves is a brand new tool that I've been developing, and it tries to address some of these major friction points that we have. So Leaves stands for Large-Scale Ecoacoustics Annotation and Visualization with Efficient Segmentation. And so it's an open source client-based web application. So this means that you just run it on your local machine. You have no need to sort of upload giant files to a cloud, and you sort of interact with it through a web browser interface. Under the hood, it's built with Python and JavaScript, and it uses a host of other scientific libraries like Librosa and scikit-learn. And importantly, one of the key reasons we sort of decided to go with client-based as opposed to server-based is that it actually does processing locally. And so what this means is that even in low bandwidth or field conditions, this tool can actually still be used. So you could take your laptop to a remote station and still use this tool, which is sort of one of the sort of driving factors behind why this tool even exists, to be honest. And so this sort of addresses that a practical concern that not every researcher has high-speed internet or a cloud computing resource in the field. So Leaves was designed to be accessible offline on modest hardware. So just briefly on some existing tools out there, because there's a ever-growing number of these, and to compare them together is quite difficult. But I tried my best in this this Leaves paper that and so I tried to sort of organize it by paid and free tools. And the sort of bottom line is that a lot of these tools are sort of fragmented, and it's a very much a patchwork landscape. There's a lot of software for different tasks. You might have one for viewing spectrograms, another for running detectors, another for clustering or annotation. So what I find is that a lot of these tools, they're great for small scale analysis, for example. They have really nice user interfaces, but they unfortunately just don't have that that part where they can't actually they don't have the features needed for large scale PAM. So a prime example that comes to mind is a lot of these tools, as far as I'm aware, they sort of treat it might be you're labeling a singular spectrogram, for example, or a singular sample, and so a lot of these sort of things obviously aren't ideal for large scale things, large scale PAM application. And even beyond that, what we've tried to do with Leaves is support efficiency tricks by including things like batch sort of uploading and these sorts of nice tools, which unfortunately just a lot of these other ones they seem to lack. But they're all great in their own merit, which is the good thing. They all have their own application. It's just that when we end up performing repetitive tasks manually or juggling between tools, it's sort of at that point where we start to wish we had something which maybe help sped up the process. And that's what Leaves really was designed to be. So here's what Leaves, the sort of this is what the interface looks like. So the core workflow of Leaves uses unsupervised clustering to sort of group together audio segments in a way to speed up annotation. So essentially, instead of listening to every minute of recording, you let the computer sort of automatically find clusters of similar sounds, and then you, the expert, step in, label those clusters or samples as a group, and then based on that, it sort of applies to the entire group. And so by labeling the sort of whole cluster in one go, you can annotate potentially hundreds of instances with the effort of a few, which is really nice. By the way, this is on GitHub for those who are interested, and there are sets of instructions on how to download it, how to use it, and all that sort of stuff. But I'll hopefully go through a little bit more in the demo. But briefly, just sort of at a high level, the Leaves workflow is designed to work with large-scale ecoacoustics datasets. So essentially, you start by uploading your raw files. This could be, several weeks. I've done an experiment with several weeks worth, and it supports common formats like WAV, MP3, FLAC, and once uploaded, the sort of system divides the audio into shorter clips. By default, 4.5 seconds, but that could be changed. And so, there are also a lot of optional settings for sort of noise reduction filtering and all those sorts of things. So once it's sort of done that, it extracts features. Here we sort of use MFCCs. It also generates visual spectrograms. So this is all sort of happening under the hood. And a lot of the sort of data gets compressed into 3D space using UMAP, which we then visualize it in the interface. And so ideally, the end result is a 3D plot where each sort of cluster ideally represents a each point represents a sound clip, and so each cluster then represents a group of sound clips which are hopefully related. And so we can then use some sampling approaches to take subsets of these clusters, say 10% of a cluster, and then have the expert listen to that 10%, label that, and then we can then sort of propagate that and have the cluster inherit that label automatically. So there's a little more to it than that, and hopefully in the sort of demo I'll be able to sort of go through it in a little bit more detail. But once you're done, everything can be exported to a CSV file that includes your annotations, your feature coordinates, metadata, all in a sort of format that's designed to work with other tools as much as possible. So when we actually when I was developing this tool, we tested this using some data from 6 of our sites across the A2 network. We selected 6 across the eastern side of Australia, which are currently under close examination by ecologists. So we also chose these because of the ecological significance and the sort of variety of ecoregions that they represent. We then ran this through our system and processed it through our tool. And so the outcome of that was that, in our evaluation across these 6 real-world data sets, which I should also just briefly mention. So we've got Reniru, Andara, Wambiana, Morochan, Duval, and Takata. I hope I'm not butchering them, but I'm pretty sure that's how you pronounce them. And then based on our those 6 data sets that we collected, we ended up using leaves to annotate them and compared it against manual labeling. And what we found is that across all 6 data sets, we're able to label the entire data set up to almost 7 times faster compared to the sort of manual traditional spectrogram annotation approach, one by one. And despite that speed up, we were still able, the labels that were produced at the end were still quite reliable, and we saw between 79 to 90% agreement with independently validated ground truth. So in other words, you can annotate much faster and still retain around 80 to 90% of accuracy, compared to manual labeling. And so this is a huge boost for projects that need to label big audio collections. And so, we hope that sort of this helps to generate high quality label data, and we can focus on answering more interesting questions rather than just sort of wrangling data. So the final thing I just want to touch on before getting to the demo is sort of the leaves use case. So when we set out to design this, we didn't really plan on getting to the species level ID at the beginning. And the reason for this is, inherent in sort of unsupervised clustering anyway, is the idea that we can sort of generally group things together at a broader taxonomic or sound type level. So rather than sort of saying, all of these sounds belong to this particular type of species, we can say that all these sounds are insect-like, or, this is all rain. And the idea is that leaves acts as a sort of pre-filtering step. to organize and reduce the data. And so once you sort of got these annotated, labeled samples, which hopefully you get rather quickly, you can then directly, use that to, you can kind of think of it as sort of a course annotation, I guess. So this helps up, obviously this can help us to assist on studies that focus on soundscape competition and sort of higher level, broader questions, I guess. But at the ultimate end of the day, Leaves helps and allows researchers to reduce time and focus on relevant subsets. So you might find in your data set clusters which aren't of real significance. That could be just full of rain or full of insect sounds, for example. And so what you can then use Leaves to sort of filter your data set. So with that, I think it's time for a live demo. So hopefully this doesn't crash and burn, but let me share my screen. So just sort of at a high level. So starting from settings, which is probably the most boring area, to be honest. But for those who are interested, there are quite a few different settings and sort of, fine tuning that you can do with sort of the different processing methods and feature extraction and complexity reduction and all these sort of things. If you're familiar, if you really will want to get into the sort of the weeds with this sort of thing, we do have those options available. But for those who are less familiar with these sort of algorithms and things, we've also got a sort of optimal one that we've found that works, at least across our 6 sites, the most optimally. So the idea is that once you've sort of got your settings down pat, I'm just going to leave mine all as default for now. The idea is that once you've sort of set your settings, you can then go ahead and upload your data set. So for the sake of brevity, I'm not going to do that, but you can upload multiple files at a time. You can do, it can be continuous, it can be in 2-hour chunks. It works. It basically, under the hood, once you upload, it sort of does all the processing and ingestion where it'll sort of chop up your file into your predefined length of segment. It'll do all the pre-processing. It'll feature extract, reduce it, using your dimension reduction. And then at the end, it'll produce a nice, hopefully, embedding that looks a little bit like this. So generally, it can take a few minutes depending on the dataset size. So for the sake of brevity, like I said, I created this prior to this, so hopefully to basically save time. But it is quick, and it's getting quicker based on algorithms as well. So on the left-hand side, you can see all of our clusters, and what we've got here is we can actually just click on individual points if you're really interested. So I'm just going to click on one. I hope the sound's coming through. So that just sounded like an insect with some water or something in the background. And so you can actually click on each of these individual 4.5-second clips to figure out, what's in there. But the way that I that I envisioned this tool being used is that you sort of sequentially go through each cluster. And so starting at sort of cluster 1, you can go through, we do also have keyboard shortcuts. And so you can go through, it will update the spectrogram and waveform. So it takes a portion of the cluster, which is decided by you. So for now, I've just set it to 10 points statically. You could do some sort of proportion, or you could do some sort of random sampling. We're still yet to decide ultimately on what we're, going to implement, but this is something that you can change in the settings. So the idea is that once you're, as you're listening to each sample, for example, if I click the next one. So I heard insects, and I think I heard like some, might have been some rain in the background. So now those labels have been associated with that sample, right? And so the idea is that once I go through all of the subset samples for this cluster, so once again, that sounds like an insect, and again, some sort of thing in the background. Once I go through all 10, however many, it will then take the majority label and apply it to the rest of the cluster. So this cluster 0, sounds to me a lot like just insects and, possibly some geophony in the background. So that will be the propagated label for all of those points. And, assuming and hopefully ideally that our clustering and all our settings are correct, and that these points are related, and then that should hopefully lead to a good result, like we showed in our paper, between 79 to 90% sort of accuracy, depending on the site. So the idea is that you can go through each cluster, listening to every to this a subsample of the cluster, label those points, and then it will propagate to the rest of it, and that way you can quickly basically label a data set. Once you're done with that, once you've finished going through each cluster, and there are nice things where you can just look at singular ones, for example, or you can exclude ones that you're not interested in. So say you're not interested in those insects, you can actually just remove it from, you can filter it out. So once you've sort of done this, you can save the annotations, download your CSV file, and then here's, I'm not sure if I'll be able to show it, but here's one I prepared earlier. If it's going to, I'm just going to switch over. So I went through prior to this session and just sort of went through and labeled probably the first 7 clusters or so. And so what you can see is, again, because I set it at 10 samples per, so here, essentially what we're seeing is XYZ are the coordinates that we use for our plotting. The class is our cluster. Sound path is where we get a sound from. These are the classes. So again, these are modifiable, by the way. So you're not limited to just these. This is easily, there's a text file, you can literally just type in whatever you like. And for every time you clicked, a 1 would show up, for example, for that particular sample. And because this was, I think, yeah, cluster 0 was mainly just a insects one, that was what our propagated class was. So I'll just briefly close on a few final notes before we get to some questions, which I'm sure some people have. So when we look at this sort of, this pipeline, like we've been saying, the way I sort of envision it is, now rather than having that sort of friction, we have this sort of shift where I envision one that integrates sort of this automation with expert oversight seamlessly. And so hopefully, this sort of helps us steer in a direction where we can start actually looking at handling large-scale ecoacoustics data sets more sort of seamlessly and with minimal friction. So looking ahead, so we acknowledge that, with segmentation and annotations being the critical sort of bottlenecks. However, now we've got some tools to help us drastically improve the situation. And so hopefully by combining, further smart algorithms with human expertise, we can turn this laborious annotation task into a more guided and manageable process. We've got some pieces of the puzzle. Things like BirdNet are really nice because, eventually it would be really great if we had a BirdNet, we had every species listed in the world, right? That would be ultimately ideal, but we have a few steps to get before we get there. And I hope that leaves can help sort of fill a major gap that we have in this workflow. Hopefully it sort of acts as maybe the first piece of glue that connects this sort of big audio data with our, our downstream interpretations and help us sort of effectively manage our data. So there's a few remaining challenges, even with leaves. For instance, species level clustering, I know a lot of people are going to be interested in this, but it's a lot more difficult to get to species level than the sort of broad categories that we've been looking at. However, it has been done, which is good news. So with similar approaches where, in current research, we have been able to get down to species level. So hopefully with future iterations, we can improve these algorithms. And hopefully another thing is improving the transferability, because it's still a little bit unknown to us whether if we apply the same clustering settings to a very different habitat, say the Australian rainforest to an African savanna, for example, will it work as well? That's still very much a unknown to us. So there may be no one size fits all. So continuing to gather these sort of case studies and perhaps enabling some more adaptive learning will help. So lastly, with unsupervised learning as well, evaluation is tricky. So even though we have ground truth, just been, developing, I guess, robust metrics to actually measure how well our unsupervised clustering is actually doing is something that we still need to work on, I think. So I hope that this talk today has given you a sense of optimism and excitement about where PAM's heading. And, the future PAM pipeline I envision is one that is efficient, scalable, intelligent, one that minimizes tedious work and maximizes insight. So hopefully by reducing this sort of friction in our workflows, we can spend more time doing science and conservation and less time spent looking at spectrograms. So I invite you all to check out Leaves on GitHub. It's freely available. And if you have any feedback or ideas that could help shape future features, I'm all ears. So thank you very much for listening. I'd just like to thank my supervisors as well, Professor Ikshai Lee, Professor Lynn Schwarzkopf, Dr. Eugene Ahn, and Dr. Slade Allen Ankins. They've been immeasurably helpful in my PhD journey. And yeah, thank you all. I appreciate your time.
Hi, Thomas. Thanks so much. I'm hoping you could see the torrent of emojis streaming in. Yeah, just incredible tool. And also your presentation was really clear, and I think will be a useful resource for people just trying to learn more about automated approaches in general. We have a lot of great questions in the chat. Before I do that, I also wanted to highlight that 2 weeks ago, we had Fran talk about his insect R package, Orthoptera, and Fran has agreed to give an office hours, basically open session for anyone who's interested in trying out this tool altogether. And so that'll be at our usual BioacousTalks time next Wednesday. And I'll throw out a reminder and announcement about that. And maybe, Thomas, we could find a time for us all to get together and all of us try out your tool.
Absolutely. Yeah. Absolutely.
Right on. Okay, so I have some of the themes from the questions. And one of them just has to do with like constraints or, with respect to frequency range or the size of the files that are being input into the tool. Can you talk more about the, yeah.
Yes, totally. So as far as file size, I don't think there's any limitations. Basically the limitation of your hard drive, because it's all sitting locally anyway. So yeah, if you've got terabytes of storage, it's all available and you can process it through Leaves. We sort of designed it to be that way where we can handle a lot of those live data sets. So as far as our frequency ranges, I don't think it'll work well for bats, I must be honest. So as far as like ultrasonic, probably not going to be as ideal. I have had some interest from people who want to use it for like whales and that sort of thing. I think it could potentially work for that, for sort of underwater species, but to be honest, I haven't had a chance to test it with that. So I'd be interested to see the results if anyone has it.
Yeah. That's two of the questions. Can leaves identify vessel noise or other clusters in underwater recordings, and in areas with high endemism or with unusual vocalizations, or with rare or infrequent calls, where maybe you just have one or two instances. What's the use case there?
Well, fantastic news is that Leaves is great for doing that. And I did some tests myself where I don't know if you know this, but koalas actually make a noise, and they don't make it often, but it's sort of like a grunting sound, and it's quite rare. So what I found is that when I've passed through a week of data from a site that had koalas, it actually sectioned it out quite well, and it was very distinct from everything else. So those call, depending on what sort of species it is, if it's something that is very distinct, then leads will do quite well in actually separating that out. But if it's all to do with the similarity of the sort of tonal sounds. So if you have like I was saying, the sort of a frog and a bird in Australia that sound pretty similar, it might group those together. That's the only thing. So
yeah, and one question that I had was, you mentioned that the accuracy is 70 to 90% compared with human level annotations. And I was wondering, like, if you have the vast majority of samples are just like background noise and insect choruses, then I could see the accuracy metric being mainly just like a reflection of those way more common sounds. But have you measured how the accuracy differs depending on class type, whether how common or rare it is, or maybe the tonal properties of the sound itself?
that's a really great question. And to be honest, I haven't investigated that. I haven't even experimented with that. But what I can say is that it's all to do with the features, right? So here we're using null frequency Sefton coefficients, and yeah, those seem to work well. So if we're able to, I'm sure there's other experiments going on out there where they are using different features to find that sort of stuff. And potentially we're looking to integrate further feature sets into Leaves beyond just what we've been using up to now. So it's all very possible.
Excellent. Does anybody else have a question for Thomas? Okay. Somebody named Glenn. Hi, Glenn. Have you explored using this tool in the context of taxonomic work? For example, can this be used to quantify the distance in multivariate soundscape between recordings made of the vocalizations of two different populations that are hypothesized to be distinct species? So maybe instead of PAM data sets, it's a curated archive of vocal recordings, and you have 1,000 from species A and 1,000 from species B.
Again, a great question. That's something I would be very interested to try and Leaves. I would expect them to separate out. Again, it depends on the similarity of the sound. So if they're two very distinct species, they'll separate out, pretty well. Again, it all sort of depends on the sort of acoustical makeup of the segments, I guess. If there's sort of an underlying, similarity between them, it could be, if it's like a similar sound of, rain, for example, then potentially it could all cluster together. In which case you would need a different feature set or different settings.
Is this tool primarily clustering Is it mainly using the spectrogram as the input?
No, it's using the raw audio.
Is that turned into an image?
Yes.
And then that's what's being used. The cepstral coefficients are being calculated on that, and then it's those that are being used to make the clusters.
Correct. Yes. The spectrogram is only for the annotation stage, I guess, where it helps to visualize it. Yeah.
Mhm. On behalf of everyone, I give you a big round of applause and just thank you for your work on this tool, and just as much for joining us here today and sharing about your work. I really appreciate the effort that you put into making this so clear and comprehensive. And
Very.
so I'll reach out to you, and we could find a time for like an open work
Yeah.
session.
Yep, definitely. Yeah, no, I really appreciate the opportunity to speak here in front of everyone. Please, if you I'd be happy to connect with anyone. If you have any questions, feel free to, that you come up with down a week down the track. Feel free to send it my way.
Oh, just at a buzzer beater by Armin. So can we handle one last question?
Yeah, absolutely.
Okay. So in approaches where clustering is used to group similar sounds, and human experts annotate clusters instead of individual recordings, doesn't the system still rely on a reference set of known species sounds for accurate labeling? And maybe you could talk about why 10%. And wouldn't it cause a species ID bias for cryptic acoustic species?
Yeah, that's a very loaded question for the buzzer. Yeah, so sort of unpack that. So no, it doesn't rely on a reference set of known species sounds, because the expert is acting as that verifier, right? So with unsupervised clustering, we're grouping together sounds that are similar, but we don't know the label, we don't know what they are. We're just doing it based on the underlying acoustical makeup, right? So that's, yeah, we don't require any labels prior. Why 10%? So we found that a lot of our clusters, sorry, were in the range of between sort of 50 to 300, 400 points or so. And so what we found is that 10% falls within a sort of statistical range that is generally acceptable for sort of subsamples, I guess. And it is a modifiable figure. If you want more fine-grained clustering, and annotations, you're absolutely free to change that, and you can go through every single one. But obviously you lose sort of that the propagation and the sort of efficiency gains. So it's totally user dependent, which is really nice. So if you want like a very general overview, you can even do less than 10%. So
Yeah.
as far as bias for cryptic acoustic species, um, species ID bias. To be honest, I'm not 100% sure what the question is asking. So
Maybe if it's, an animal that produces a very short, or, there's a lot of overlap, it's tough to distinguish. It doesn't have as much charisma as the other sounds. It might be harder to form distinct clusters where that sound is.
yeah, yeah. Yeah, and we're absolutely working on that. I've got an upcoming research paper where I'll be looking specifically at the sort of robust distinction in terms of overlapping frequency and also time. So be on the lookout for that.
Nice. It just occurred to me how nice it would be to have a collection of benchmark data sets from around the world that we can use, so that a tool that was developed and tested in, across some habitats in Australia, we could then have like a pretty systematic way of assessing its performance and how it differs across different biomes and domains. So if anyone wants to take on that project, I think it would certainly be helpful for tools like this.
Yeah, the problem is that there's a lot of tools popping up these days, so you'd have to figure out what the best one is and then do it for that. And if there's a new tool coming in every other month, then that's a lot
Yeah.
of repetition again, and you're back to
Yeah, but
this
like,
back to problem.
what if you just had one day per biome or per site that was comprehensively annotated, or 1 minute per hour for a day at all these different sites, and that was just put up online? Oh, Geo just shared a data set that could be exactly what we're talking. Yeah, I just saw this come out. Cool. This I think would be a good BioacousTalks as well. Yeah, so at least we'll get an office hours and people can see how the tool works in all these different parts of the world.
Yeah, yeah, sounds great.
All right, well, have a great rest of your morning, and we'll be in touch.
All right, great. Thank you, everyone. Thank
All
you,
right, thanks,
Ben.
everybody. Bye bye.
Cheers.